All right, welcome everybody. I hope you all are enjoying your lunch. We have a bit of a departure from the norm here. Instead of lawyers talking to other lawyers about the law, here at the IP and Biosciences conference, we thought we would actually invite somebody who's doing some really fascinating cutting-edge biosciences to talk about what he's doing. I think as you'll see the talk, you'll see that it's going to have some really interesting implications for the law. It's also going to have some really interesting implications for our world. In a lot of ways, Drew Endy, who's in the bioengineering department here at Stanford, is probably the leading person in the emerging field of synthetic biology. If you don't know what synthetic biology is, you are in for a treat. And he's going to tell us a little bit about turning biology from a craft into an engineering discipline. So please welcome Drew Endy.
Can you hear me okay? Okay. The real honest disclosure for me is the title of this talk. That's what I'm excited about. That's what I do. As a way of quick background and in front of judges, to give you a sense of the pitfalls I've run into, experiences I'm connected up in, I have been fortunate to work with the Committee on Science, Technology, Law back in DC, the folks that shipped this book, Reference Manual on Scientific Evidence. I have nothing to do with this, but my name's somehow in the front matter. I'm supposed to be rotating on to the National Science Advisory Board for Biosecurity, which has been in a kerfluffle recently around the flu work done in the Netherlands, but I'm not actually on that at the moment. I'm part of a public benefit charity called the BioBricks Foundation, which among other things has drafted a bilateral contract system for contributing to the public domain uses of genetically encoded functions. That work was led by Mark Fisher, who's a trademark and copyright attorney out of Duane Morris in Boston, one of the early drafters of the GNU public Emacs license. Excuse me, it was that long ago for Stallman. I am a co-founder and director of a DNA synthesis company called Gen9. I have been advising the Sloan Foundation on policy investments, and I'm part of a thing based out of Cal up in Berkeley called SynBerk, which is an NSF project to advance the engineering of biology. So it's the Synthetic Biology Engineering Research Center. I'm mostly keen on supporting and strengthening the public domain in biotechnology, but I'll also admit I'm in the middle of filing a patent on something, so it's a complicated spot. I wanted to start the remarks by giving an example of something we've worked on for the last 3 years in the lab here at Stanford, so you get a sense of who I am and what we do, and then I'll zoom out and give you a meta context, and I'll wrap up by bringing us back to property rights and puzzles and opportunities with respect to ownership, sharing, and innovation frameworks. If I were to I'm sure you've heard this before, that property rights is rate limiting oftentimes in biotechnology. I'll go out on a modest limb and say that's really going to be true, given the path we're on right now, and I hope you'll take from the section of the talk when I outline how the process of engineering biology is changing, what the collisions look like, where the stress points are, and what the struggles and opportunities might be. Before I get into this any further, I want to acknowledge the special nature of this venue for me. I moved to Stanford from MIT. There's wonderful law schools in the Cambridge and Boston area, but it's not the same as having a law school on the campus. And so, Mark, especially I'd like to thank you, your colleagues in the law school. Linda Kale, a colleague who is working at Stanford, jointly sponsored between engineering and law, as we try and figure some of this out. It's been an amazing place to find an intersection and work together on what the puzzles are. Okay, so if I put on my nerd hat and go into the laboratory, the thing I typically say to my team is I want 8 biobits, biological bits. I want data storage that operates inside living systems. I want to put a byte of data storage in every cell in your body. All right, so when you think about data storage, you usually think about a USB stick that might have a gig of data storage. I want 1 bit or 8 bytes. So a very modest amount. I don't need very much data storage because I only want to do things like count cell division events, see how old your cells are, begin to reprogram them as they get too old or go haywire. So how do we do this? We do this using the tools of genetic engineering that have been pioneered and established over the last generation, the last 35 years. And we use those tools to do the following. We take a chromosome, double-stranded piece of DNA inside your cells, and we engineer a specific sequence on that chromosome, which is a couple hundred bases of DNA. And we're going to call this thing the flippy in private, but now you get to hear that here. And we're going to take this sequence of DNA and hope to flip it back and forth. And as we flip it back and forth, we can use that to store information. So for example, if I had genes on either side of this element, this data register, if I flip it pointing left and turn on that gene, maybe I could call that a 1. Or if I flip it back and turn on the right gene, the green gene, call that a 0. So now I've established a binary digit, or a bit, in a chromosome. That's defined by the orientation of a sequence of DNA within that molecule. How do we do this? We take enzymes from natural sources, we express them in a cell, cause the flipping event to happen. That's writing information in. And this is what it looks like physically. I have cells down at the bottom which are glowing green because they're in the 0 state. I have an input signal that comes in. The cells switch from being green to red. The input signal goes away. This is very important. And the cells stay red. They're storing a memory of the signal, right? So that's data storage. We can create one memory, one bit. Then I need to be able to flip it back, to reset it. And the reason this is important technically is if I can only flip in one direction, then I can only do things like implement a thermometer where the temperature goes up, 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 up. But if I can flip and flip back, then I can implement what's called combinatorial data storage. So if I have n registers, I can implement 2 to the n bits of data storage. All right, how does this work? You got to express the same enzyme, but then tweak it with a partner that reverses its function, and it causes the reset reaction, restoring the element. Cells start in the red position, you give the reset signal, they become green, and they mostly stay green. So you can kind of see, it's a little bit washed out. In the middle of that right bottom picture is a red cell. It's not perfect, but it's pretty good. We can mostly reset. Now that was done 3 years ago, right as we had set up the lab. It only took about 1/4, 3 months of work. Then we needed to put the set and reset together to implement a combinatorial full bit data register. And this created havoc inside the cell because we didn't really know how to balance the levels of proteins to implement these competing set and reset operations. For example, if I'm running a reset and the excisionase protein degrades too quickly, my reset all of a sudden becomes a set. I know these are details, but I just want to give you a flavor for what we work with. So I fix that by making a lot of this protein and stabilizing it so it's not degraded, and we have to get methods for doing that. That works, except now I have a cassette that makes that modifying protein that's so strong, it's on all the time, just randomly, because cells are noisy. They're always a little bit of molecular diversity, and that messes up the set operation. My set operation now becomes a reset. So we work, and we work, and we work, and we work, and we work on this, and it's mostly artwork, craft work, manual labor, done by experts. And the reason I think this is, after 17 years of research experience, is what we're working with when we do genetic engineering typically are found objects, natural genetic elements that have been partially repurposed to implement some engineered genetic program. And that means they're all a little bit weird, all a little bit different, such that when I put them together, their behavior is not like snapping together Legos. Their behavior is like making a custom work of art that you have to test empirically every time you try something new. It'd be like trying to build the Washington National Cathedral from found objects, rocks like this. Thus, it really does take us 3 years and 750 attempts through a design-build-test cycle, meaning 750 different DNA designs, just to combine a set and reset function into one cell. And when we produce that artifact, it's a work of art. There's all sorts of weird peculiarities. If you want to talk about details, the start codon isn't ATG, it's GTG, because we needed to tweak that. And anyway, it works. So this is what it looks like when cells are switching between states, green, red, and holding state. This is probably the world's best genetically encoded data storage element on the planet today, which isn't saying very much, because there's not a market for genetic data storage yet, but probably in 20 or 30 years, there'll be a big market. Great. So that's my life as an academic. We do this work. We get money to do this work. The paper's coming out next month. Now you know about it ahead of time, so you can talk about biobits when you hear it. OK. Here's the pop quiz, though. that'll bring me into the meat and potatoes and then ultimately property rights and innovation. Why is this the case? Why is it that one human generation after genetic engineering is invented, after the original recombinant DNA methods patents have expired, that I still have to do all this? See, I grew up in Pennsylvania and studied civil engineering. I worked for Amtrak one summer. I'm familiar with building things. I'm familiar with building things where we have tools that make the building of things easy. So I'm really puzzled why, since I'm born when genetic engineering is invented and then come to it much, much later, why there's still so much research? I just want to express 3 genes. So how come? I don't really know, so I want to give you this quiz and you can help me figure it out. And then I'll share with you some experiences. Maybe nobody's been working on making it better. A lot of people have been working on some tools, but mostly on applications. Cure my disease, save the environment. And that could be because the business systems and the political systems want answers immediately. Like, cure my disease, please. Or when will that drop-in biofuel be ready to go to market? Or maybe it's impossible. Biology could be a type of physical material, it's living material, and we're never going to get good at engineering it. Or some other reason. I actually don't know, and it could be simply that there's an operational answer here. What I experience, however, is A and B. And so let me give you anecdotes of experience. For example, when I was at MIT, we were implementing the first new undergraduate major in engineering at MIT in about 30 years. It was called biological engineering. And so good engineers, when you need to do something new, you see if anybody else has already done it, and you steal from them. Okay? And so I found one of my graduate students who had earned a degree in bioengineering, and I borrowed his introductory textbook, thinking that we'd get some good ideas. And as I read the preface, I got to the 3rd sentence and encountered what you see here. So apparently there's something called bioengineering, and this is a bioengineering textbook in use at a top 3 engineering college in the United States at the time. We're going to exclude genetic engineering. There's a lot of good stuff in this textbook about medical imaging, artificial joints, radiology, right? What you might typically think of as engineers working with biology. But at the cellular, genetic, molecular level, So maybe not all of the engineering community has really engaged yet with biology as a substrate for making things. Here's another example. I was before the Energy and Commerce Commission back in 2010 when there was a kerfluffle around somebody synthesizing a genome and then is that a new life form or what's going on? And the Honorable Joe Barton, the ranking Republican from Texas, asked, could you engineer a genome that would predispose folks to vote Republican? This is a really good question, right? Because if the answer is yes, there's all sorts of follow-up conversations that need to take place. But I give this as a funny example for how when people turn to biology as a partner, they really do have needs. And this is a weird type of need. But curing a disease is not a weird need. It's a very pressing need. And what this means is the teams that get assembled to tend to work on biotechnology applications are oftentimes overdriven by the immediacy, the human story associated with the application or the environmental story. So I want to zoom out. I'll start with my roots and then switch to electronics and come back to bio. I want to give you stories of what it means to make a type of natural material easy to engineer. And the purpose of sharing these stories is so that you might help me and others steal lessons for how to do it, and then we'll try it with life. So stone. All right, this is the easy one because we're more familiar with it. We begin to engineer with stone from found objects, like these rocks. Some people become so excited about the differences of rocks, they study that. They become geologists. But if geology were a science starting today, it wouldn't be called geology. It'd be called the rockiome, the study of all the rocks on the planet. But other people like to build things, and eventually, after a while, it becomes frustrating to build with irregular objects. And so what happens is the builders invest energy, it looks like, in regularizing the material, making simpler objects. And as you make simpler objects, that allows for standardization, which results in coordination of labor across time and place. Somebody could make the rock in the quarry, another person could assemble the rock on site. This aqueduct in Segovia was made 2,000 years ago. Others continue to become frustrated with material, so we grind up rock and we make synthetic rock. You don't think of concrete as synthetic rock, but that's what it is. And if you walk around the main quad here, Stanford was so proud of the paving around the main quad that stamped in it is the branding. It's somebody's name, artificial stone. Right? So about 100 years ago, synthetic rock was so cool, it was part of the marketing strategy. And when you make artificial rock and you have a lot of other capacities, you make still more improbable artifacts, like this viaduct in France, or a city like Hong Kong rebuilding itself in a very short period of time. So zooming out, what are the lessons from our Stone Age, which we inherit and occupy now? Invest energy to regularize material, that sounds neat. Get better at assembling things and composing them so they work. Use standards to coordinate labor across time and place. So somebody in Australia could help me with my data storage project here, but I don't need to actually have a conversation with them. They just help me. We're going to need a lot more tools. Maybe there's others, and you could email me or talk to me afterwards. Now, what about electronics? This is a more modern story. Here's a computer, circa 52. It's helping to design nuclear weapons. Great. This is a computer 25 years later. Right? How did that happen? And this is a computer today, right? The web, whatever that is, the cloud. So how can we explain that geometric increase in computing engineering? Digging into one point in time, and I picked this point in time because this is the point in time that recombinant DNA is invented, so mid-70s. Here are photographs of human beings engineering computers. They must be doing very important stuff. These ladies are probably making the mask that's used in the lithography process to establish the pattern on the silicon wafer. You would have a hard time taking these photographs today unless you went to a reenactment of computer engineering from the 1970s. What you find instead is the websites for companies like Synopsys or Cadence Design Systems that do billions of dollars in sales for tools that automate the process of designing and constructing computers. So over the period of time when genetic engineering was invented to when I show up, I don't see the same change. I still see expert manual labor doing the design, build, test cycle. Whereas in electronics, I've got radical transformation and birth of new tools industries, automating and accelerating the process of designing of machines, computing machines. Why is this? Where can we get to the lesson? So here's my attempt at it. Metrology, the science of measurement, Shannon's information theory, languages for programming computing machines, standardization, the transistor logic data book from TI, fabrication of silicon wafers, computer design, electronic examination, reverse engineering, control and dynamical systems theory. How many of you think about these fundamental capacities in engineering when you go to the Apple Store or the Dell website and you buy your next computer? I don't. So I can speak for myself and say, I never think about the investments in silicon wafer manufacturing when I go to buy my next computer. I never think about who made C++. Right? What I think about is the immediacy of the application of the technology. Can I send a text message with an attached photograph to my mother? Right? And if that's true and I need to do that, then I'll buy the machine. But the explanatory power of that alone doesn't suffice if you need to go to 1952 to today and figure out how did we get to where we are. You have to go underneath and say, we sustained investments in tools for decades that made incremental improvements in the design, build, test cycle. That's at the core of engineering. And as a result of making sustained incremental improvements in tools capacities, we got geometrically better at making artifacts. That's the hypothesis. Okay, so I don't want to get hung up on metaphor. You could reject what I just said, that'd be fine. But just to get you thinking and sharing and then turn it to biology. Could we make biology, living systems, easy to engineer? Not to replace the jungle, but just to partner with it and learn from it in very powerful new ways. We haven't seen geometric improvements in biotech thus far. It might be impossible. That was one of the options. Why? It's a different type of physical stuff. And this is technical, but just to air it out. Inside a cell is a self-mixing molecular system, which is dominated by thermal noise, Brownian motion. These systems copy themselves directly. We're all familiar with this. This system does not, bud and make two iPhones yet. The systems are very high heterogeneity. So it's not like a chip that's got billions of transistors. We've got an organism that has many different types of cells. Within each cell, we have tens of thousands of different types of biochemicals. They have different functions depending on the weirdness of how they're decorated with modifications. I don't know how to engineer that. And oh, by the way, if we actually make any progress on getting better at engineering biology, everybody's going to freak out. The living ethics ramifications are at the forefront. Okay, well, we're doing it. So let me get to this quickly and give you the context. The top row shows the dominant tools for the first gen of genetic engineering biotech. Recombinant DNA for cutting and pasting, PCR for amplifying, making single edits, or a few edits at a time, sequencing for reading out what you've done. The tools I want to introduce you to, one of is old chemistry, but now it's being industrialized, DNA synthesis. The second is borrowed from computer science called abstraction, which is a tool for managing complexity. And a third area of tools work is in standardization for coordination of labor. There's probably other categories, but they haven't emerged yet. This work has really only come together over the last decade. And in fact, for example, just yesterday, the White House, I think for the first time ever, released a blueprint for the bioeconomy in the United States. And it begins to get us on a national strategy for doing this type of work. But if you read the New York Times description of that, Phil Sharp from MIT is going, you know what, this is actually the first time I've ever seen executive leadership around biotechnology at a national level. Right, so it's an interesting point in time where a lot of things have begun to gel, but we're not really full steam yet. Here's DNA synthesis. These 4 bottles, taken a photograph in my office over there, are phosphoramidites. Khurana and others decades ago did miraculous synthetic chemistry to figure out how to establish raw materials that could be transformed into the bases of DNA such that you can synthesize from scratch new genetic material. So if you're familiar with a 3D printer or a laser printer that produces an object, we have DNA printers. 4 bottles, you can guess there's 4 bases, A, T, C, G. The chemicals are derived from sugarcane, so it's a commodity substrate to begin with. They cost about $250 each. So it's $1,000 of material. And there's enough material in these jars to make 30 copies of every human's genome on the planet today. That's a lot. We don't have DNA synthesizers that can print human genomes today. What we have are machines that are sometimes clunky, sometimes they're more sophisticated, like at Agilent Laboratories, they use inkjet printers to do this. But what these machines do, they're called DNA synthesizers. They take information, this is key, this is one of the big deals here, they take information coming from a computer database or a network, and they plug it into this machine, and the information specifies the sequence of DNA you want. That then controls the actuation of the plumbing, the valving, so that the bases are sputtered out and connected in the order you want. You then get the physical material, and if you can put it back in a cell, you now have that cell with that DNA inside it. DNA synthesis is, with bias and apology, the most important technology on the planet, because it allows you to go from abstract information to physical, living, DNA-encoded phenotypes. It's at least as important as silicon wafer manufacturing, would be my backstop position. But it's probably more important. In 2004, the biggest piece of DNA anybody had ever made from scratch was 10,000 bases or letters. Not too much, about a bacterial virus maybe, a small one. 2 years ago, though, it was 1 million, a megabase. That's enough. It got a lot of attention. It was a bacterial chromosome. That's a 100-fold improvement over 6 years. That sounds pretty good to me. That's geometric, right? So what would another 100-fold improvement look like this decade? 2016, 2020. We'd see somebody reporting on a 100 megabase construction project. 100 megabases is a lot. So that's every microbe's genome I know about. Not all together, but one at a time. It's enough for baker's yeast. Right? That's less than 15 megabases. It's almost enough for a worm's genetic complement or a fly. It's just under the average length of a human chromosome, which is 128, 130 million base pairs. So it's interesting, and there's a lot of uncertainty when you try and project forward when you have geometric improvements in tools, because a little bit of uncertainty can lead to a lot of variation in terms of if and when it shows up. But I'm not going to be surprised, and I would encourage you to at least consider not being surprised if by the year 2020, somebody shows up and says, yep, I've made 100 megabases. Why is this interesting? Why does this become to be interesting? I want to give you the abstract cartoon version of this and then point out a couple things, but the abstract version should be more important because you can figure out why it really matters. This is how life works in terms of generational direct descent, replication with error, diversity, selection, evolution. What's happening is a combination of tools, particularly sequencing of DNA, which allows you at a point in a lineage, at one generation, to decode some of the information, the genetic information, to abstract information on a computer. That exists, and that's getting geometrically better. And then synthesis allows you to take that information, recompile the material, and if you can establish that in a replicating lineage, you actually now have a replicating lineage. So sequencing and synthesis technologies together make genetic material and genetic information fungible or interconvertible. Genetic material equals genetic information when you combine these two technologies. This has been demonstrated for virus genomes and one microbe genome and for lots of other subgenome systems. And it probably just gets geometrically more powerful. When you decouple matter and information, this obviously stresses and strains all sorts of cultural and political relationships. Think about what happened in entertainment when entertainment media got digitized and then decoupled from the physical substrate. Right? So you go to compact discs and DVDs, you still have Netflix shipping stuff. But as soon as you get people sharing MP3 files, right? You've coded it in information, you put it over the networks, it recompiles in the iPod locally. That's a real stress and strain on business models, on property rights frameworks. We're going through the same transition with genetic stuff. Material transfer agreements, I never sign one. Haven't for 5 or 8 years. Why? If you want me to sign an MTA, it's actually easier for me to pay somebody to build the DNA. I can ignore your material transfer agreement, as a simple example. The freak out example is, what happens if I get the genome sequence for a pathogen? Do I put that on the internet? Right? So decoupling of matter and information from these lineages is a big deal. Okay, abstraction. Quick primer on this. Information encoded on genetic material is really complicated. So TA, ATA, CGA, CTC, ACT, ATA, GGG, AGA. What's that? Right, you can memorize these sequences by making them your login passwords. Right? Works really well. But it's tedious. And yet, this is how, I'm teaching the sophomore bioengineering lab today. We were talking about base pairs in class just before and when I get back to lab afterwards. And programming at this level really is tedious. Right? Imagine if sending email required working in machine language. That could be good or bad. You might have less spam, but you wouldn't be able to do much else. So, when we get to genome scale engineering projects, here's 40,000 bases of DNA as sequence information. We're really not going to be engineering it at this level if we hope to really pull this off. Let me say it a different way. This is an example of a gap if you're hiking, a risk-taking hiker, about 4 meters. There's a bigger gap in genetic engineering. On the left is how biotechnology tends to be commercialized today from a capacity perspective. You typically have projects that are tens of thousands of base pairs of designer DNA with a dozen or so DNA components. So make lots of insulin, make lots of this fuel. But on the right side, through synthesis and other tools, we're now able to compile genomes from scratch. These aren't yet designer genomes. These are recapitulations of natural sequences. Genomes are megabases. The biggest assembly project right now is about 8 megabases. Those are thousands of genetically encoded functions in single systems. If I take 8 million divided by 20,000 as just one point of reference, that's a factor of 400. That's the biointegration gap. If you could teach me how to become 400 times better at putting the molecules of life back together, we would instantly design at genome scale. It's a very different world for biotech. It's a world where, for example, if you have tens of thousands of characters in the English language, you could write a presidential address, a New York Times editorial, a research paper. But if you let me code in megabases, in millions of characters, I'll write French plays, I'll come up with American novels, I'll come up with Russian novels. Megabases of coding capacity is now accessible from a construction perspective. The challenge is to become that good at designing. The tool is abstraction. This is lifted from computer science. The idea is you create layers of function from primary DNA sequences all the way up to high order capacities. Like, I'd like a counter that can count up to 256. And if we do this right, bioengineers working up at this high level wouldn't need to know that DNA is made up of 4 bases. They just call down for the latches, the bit registers. Somebody else would make the DNA. They get it, it would work. Much like I don't really understand machine language. Now, this is a very controversial technical topic because it is really difficult to make what's shown on this slide true. For example, when I was telling you we took 750 attempts to make a latch, that's going from this level here to this level here 750 times, because the composition of the lower level objects isn't reliable, so we have to test it time and time again. That's why people think this might never come true. Well, guess what? It turns out that we've just made it true for one very key important part of genetic engineering. What we've done is we've taken control over the central dogma, DNA, RNA, protein, and we've figured out how to engineer the componentry, the control elements, such that the compositional process is quite reliable. We did this with public benefit funding from companies and the National Science Foundation, and we're giving all of the work product into the public domain as the core for a language or operating system for programming at genome scale. So I'll just give you a version of this for people who want to see technical evidence. Across here are 22 different translation controllers and about 14 different transcription controllers expressing one gene, green fluorescent protein. I take the same control elements and I express a different gene that has a very different sequence, and the pattern is approximately identical. There are outliers comparing these two heat maps, like this point here. It turns out that's an error in the laboratory where somebody mispipetted the strain in the 384 well plate. So our composition of the control elements is now so reliable, we're finding failure in other laboratory practice. I'll say this differently. If people do genetic engineering with elements that have never been regularized and engineered to support composition, this is an example of what you get. I take two different proteins, red and green. I use the same control elements over and over again. Each point represents the same pairing of control elements. One pairing produces 60 times more green than red protein. Another pairing produces 20 times more red than green. That's typically what people do. That's why we had to make 750 attempts. I use the newly engineered regularized elements that support composition. I get this scatter plot. 93% of the things are hitting a factor of 2 window. To translate this into something that might be a little more understandable, if you're dealing with the compositional reliability on the left, you have to design and build and test systems one gene at a time. If you have the compositional reliability on the right, we can now leap ahead and design and build and test systems 10 genes at a time, because our failure rate's been reduced. Nobody really knows how to design 10-gene systems yet, so that's going to have to catch up next. Okay, so now let me turn the corner and wrap this up in the next 5 minutes, and then we can talk about whatever you like. As we've gotten better in going through this design, build, test cycle, the people who have taught us what to do and then immediately benefited tend to be the next generation of would-be engineers of biology. So here is an example. Back in 2003, I was teaching 16 students, and they spent a month trying to design genetically encoded clocks. None of that worked, except we got $80,000 from DARPA to pay for DNA synthesis, so these students didn't have to do any recombinant DNA. And when I spent that money, we figured out how to decouple design and fabrication with a professional DNA synthesis team. And it took us about 7 months after the class ended to actually make that work. That was so great that we taught the course again the next year. And we got all the DNA back from the students' designs within about 2 weeks. And none of their projects worked. This was so great that we didn't even wait another year. We taught the course that summer at 5 different universities in the US. And it was now called the Synthetic Biology Competition. And one of the 5 projects worked. Laura and Jeff in the front. Laura's now a finishing PhD student in biophysics at UCSF, and Jeff is a professor at Rice. They were able to take photographs using bacteria that were programmed to be responsive to light. They got a paper in Nature. They're doing good. So a 20% project success rate was really terrific. So you could guess what happened. More people wanted to play, and it became something known as iGEM, the International Genetically Engineered Machines Competition, or a Genetic Engineering Olympics for undergraduates. 13 schools, 3 outside the US. That was kind of fun. Got a little bit bigger the next year, about 40 schools. The students became self-aware. They made a world map of where they were coming from. They identified regions that didn't have participation. Thus, they went on trips, exchange trips. The students in China, for example, then invited us to come over, give a weekend tutorial. I met my wife. She was at Caltech. I was at MIT. This is all good. We're both here now. Peking University showed up the next year. They won. Got a little bit bigger. Got a little bit bigger. Got a little bit bigger. I can't show you last year because it doesn't fit in one room. It went to regionals. Hong Kong, Amsterdam, somewhere else. Here's an example of what these students can now do. This is actually 3 years old. These are 7 strains of E. coli that are engineered to implement distinct biosynthetic pathways to make pigments. Red on the left, purple on the right. They did this with 8 teenagers, 16 weeks, $25,000 in capital. How did they do it technically? It's a success story of do it together biotech. Half of the genetic elements that are needed to implement these pathways they got for free from a collection that the students build over time. There's now a registry of standard biological parts. When you finish this competition, you contribute your parts, and then when you start the next year's competition, each team gets the aggregate collection from all of the preceding years. Here's an example of what that looks like. Back in 2005, is a 3D 4-well plate with hundreds of genetic parts. Today, it looks like a computer database. They still ship materials. They still ship about 1 million parts a year, but the real shipment is the web API that you can then hook up to a DNA synthesizer. What would you do with living pigments? It doesn't really matter. It just sort of demonstrates that you can do this quickly, but they were able to get this to work fast enough that they connected up with the Royal College of Art and the designers there, and they started imagining what future consumer products would be based on living pigments. One of those is shown here. You would have probiotics that are also implemented with genetically encoded sensors. You eat them. They detect the disease state of your alimentary canal such that what comes out from you is a different color depending on which specialist you should go see. Probably better than colonoscopies. Right? What you do with living pigments, I don't know, but it's evidence that things are changing in weird ways. What else do you do when you can synthesize with biology? Here's a different project, fun to share with after lunch. Sissel is an artist. She works on perfumes in Berlin. Christina, not shown here, is a microbiologist at Harvard, have moved recently to UCLA. We paid for them to spend 1 month together in the lab at Harvard and 1 month together in the studio in Berlin to see what they would do, because we would like to explore if the future of biotechnology is not merely about the industrialization of nature, but leads to new schools of design, better understanding of life, new modes of engineering. So Sissel shows up and says, what's new in microbiome? And Christina says, people are sequencing the microbes that live on our skin, and what's on the outside of my elbow is different from the inside of my elbow, but its left and right outside are similar. What are you excited about? Oh, I'm excited about the odors of cheese. Cool. Those of you who have been through Boston or live there know that there's an amazing cheese shop called Formaggio's, so they got a special tour of that. They went to the aging cave. They brought a rack of test tubes. They pulled core samples out of the stinkiest cheeses and brought them back to the laboratory, where they started to do determinative bacteriology. Great. As they're talking about this work, though, they have interesting conversations like, cheeses are made with microbes in part, and some of these artisanal cheeses are made in very small batches. What is the relationship between the microbes that live on human skin and the microbes that make our cheese? Don't know. If you were a scientist, the way you would approach that is you'd start sequencing everything, and you'd do the alignments and study it. But if you're an engineer, what you do is a little bit different. You go to the farm, and you get raw milk, then you'd walk around Harvard, and you'd collect microbial samples from the skin of your colleagues, and you'd go to the incubator, and you'd start making cheese. So I happened to be going through Boston, and they came out with a tray of 48 cheeses made from human skin microbes, from Daisy's armpit microbes, from a philosopher's toe microbial consortium, from Christina's hand, from Sissel's nose, and so on. This threw me for a little bit of a loop. Daisy's armpit cheese is stunning. It is a floral citrus bouquet. Philosopher toe cheese, I can assure you, need to invent a new adjective to describe how wrong it is. But more interestingly, what this then led to was a whole bunch of questions, not just about safety, but about things like, my mom said, eat well because you are what you eat. But what this project implies is that we eat what we are. And then that night when I was having dinner with a banker who manages a group at Fidelity, and I thought this would disrupt the meal, he instead said, billion-dollar markets. Celebrity cheese. Right? So Barack Obama blue, right? Or Romney red of some form, or Lady Gaga goat, I don't know. But you have seen perhaps the trademark disputes out of London around the Gaga brand of ice cream. Okay. Now, wrapping this up, it's not just about technology, though, and I really mean this. When you get into this world, the work in the laboratory actually isn't rate limiting. The human elements beyond the bench are what are rate limiting. And here are the questions that are activated at the moment for me and many others. We're dealing with a generational renewal in biotechnology. Who's going to lead biosafety in the future? The people who figured it out in '75 are retiring in emeritus today. What's our strategy for biosecurity? Really? Who has ownership of that? How much can we make in partnership with biology? Biology is not an infinite resource. There's only so much photosynthesis. There's only so much carbon and nitrogen cycling. What's the best relationship between private and common wealth, ownership of capacity? Who chooses what gets worked on? Who's represented in the process? Who has access to these capacities? How do we make sure this isn't just merely industrialization of life, but is actually quite beautiful and creative? And how do we work together on this? My self-assessment as I wrestle with these topics in terms of degree of difficulty, if you want to think about this like a gymnast, here are the rankings. Biosecurity is really a puzzle. The best balance of wealth and property rights is probably right in the middle. It's a complicated time. So I want to close with two slides. This slide is one that speaks directly to property rights. I'm going to try it this way. Again, thanks to Linda and others for helping me improve my thinking here and doing a lot of this work. Here are the tools categories I was talking about: synthesis, abstraction, standards. What are the impacts on the process of engineering biology? What are the property rights puzzles that emerge, and what are the opportunities, if any? Well, here are some highlights to remember. First, genetic information and genetic material are the same thing, as sequencing and synthesis exist together and get geometrically better over time. I really am just at a loss to figure out how to react to all the cases that are percolating in biotech property rights right now, because it doesn't seem to appreciate this fungibility and the scaling of power in the tools that make this true. There's a geometric increase in the scale of systems being engineered. So it's not just short declarative statements, make lots of insulin. It's much more complicated, rich genetic programming emerging. And we have, through standardization, coordination of labor over time and place. That leads to the creation of new types of intermediaries, which become focal points for liability associated with property rights. For example, the puzzles. The collection that supports the iGEM activity has over 10,000 genetically encoded functions. Who owns the rights to use them? Quick, do the FTO. Have that benefit industry, not just academics. It really is amazing capacity to do things for cheaper capital costs. These intermediaries, whether it's a parts collection or a DNA synthesis company, are coming under scrutiny from the holders of traditional use claims, if you will, uses of GFP and so on. The public domain is incredibly weak in genetic engineering. It's very hard to establish something as being free to use. Please make that easier, right? So here are two opportunities. I just said one of them, but I'll restate it in a minute. But the first one, which we're thinking a lot about, is how we might shield intermediaries that provide services in DNA synthesis, assembly, or distribution from liability for patent infringement, and do this in a way that doesn't create disincentives around establishing property rights for the inventors. But this is a big deal. We actually do have letters coming in, threats being made in industry, in research organizations, and moreover, we have the opportunity cost of some of these new methods not translating into industry because nobody can figure out how to clear up things. And then second, restating, enable the public domain or legal equivalents thereof so that we can balance and benefit more directly from property rights. If you think about the maturity as it is today of computer programming, 12 of the 15 most used computer programming languages are free to use. Didn't start out that way, but it got there. And I think most successful languages actually have to be free to use at some point in their maturity. right? So we're heading towards this future where we're going to have a language for programming in life, and the successful versions of that, in part, have to be wrapped up or grounded or anchored, not just in strong property rights, but the public domain version, I think. All right, so make biology easy to engineer. Tools are probably secret. Let's foreground that. One of the things that comes from doing this work is we learn a lot about how life works. Learning by building is incredibly powerful. We figured out how to pull this off. We probably remake our civilization. How we manufacture things is going to be a lot different. I didn't go into that in great detail, but the capacity for biology to make stuff is ridiculous. And we haven't tapped into most of that. And if we can figure out how to do that well, we'll have a better civilization too. So I hope you enjoyed the remarks and might help with these challenges and opportunities. You've got time. It looks like we have about 12 minutes for questions if anybody wants.
Yes, and we have microphones here, so just head to the microphones.
Seems to me you've put up a very strange paradox, I'm going to call it. We're studying patent law. Patent law, as I understand it, requires the publication of how to do it. In this week's Economist, they published an article saying, but we have ways to do it, which are easy, public domain, et cetera, for mechanical things. And certainly you're putting together biological things. It seems to me that what we're heading toward is the same catastrophe as with the music industry, where the technology makes it impossible to stop individuals from violating everything we have to protect innovation and patents. Is this what you see in 10 years?
That's one way to phrase it. So it's a good question. We didn't have a sales and marketing program for iGEM, but practically what that resulted in was a reworking of the genetic engineering and bioengineering curriculums in the world's top 200 research universities in a bottom-up fashion. How come that worked? One of the things that's happening is there's another set of people showing up. They're matriculating into universities. Biology is incredibly cool. It's the stuff of life, so you know it's important. It's a nanotechnology that works. It's already taken over the planet. So green goo, right, as Whitesides would say. And you can think about programming it kind of with DNA, and you can certainly think about getting a lot better. So this is sort of you get that for free, that generational excitement and attraction. So is that a bug or a feature as that then connects with improved tools and capacities? For example, the interchangeability of matter and information for genetic stuff. Don't know. I'd like to say the reason I'm here is let's figure out how to make it a feature, because it's happening. I think, I mean it honestly. Those puzzles are real puzzles, right? And if you see it as a threat, that's one possible framing for sure. If you're a product catalog company today that's backstopped by 3,000 patent claims on uses of genetically encoded functions, your business model probably needs to adapt, right, to a world where people are compiling things locally, right? So you turn that either into a business advantage or a business threat, right? I think there's a lot of opportunities right now associated with the fact that you've got a literal generational renewal coming up through biotech and a lot of engagement between different technology communities who are excited to think about how to make this work better.
Thanks, fascinating and exciting. In developing World University and School, like Wikipedia with MIT OpenCourseware, which is Creative Commons licensed, we would like to have a law school in every country, around 200 countries, potentially defining patent law, but also we have all these MIT sort of subjects, biotechnology. They're a wiki, but also with many students, it would be potentially interesting to create a body of research that spanned many, many national boundaries. I'm curious about your experience in biotechnology patenting vis-a-vis Creative Commons law, a relatively new form of law, and how those two bodies of law, patent law and Creative Commons licensing, might articulate to both create a kind of open field for these building blocks that you're developing, plus, and it may not be your question as an engineer, but Creative Commons law, patent law, and the building blocks?
Yeah, so by Creative Commons law, you mean copyright law, right, and a little bit of public domain, right, and so that's fantastic work. The issue is, what's the role of copyright, if any, around DNA sequences and the uses thereof? Naively, initially coming into this, it's immediately attractive because the transaction cost associated with establishing copyright is tasty compared to a patent claim. But holy cow, as you get into it, it's horrifying, right? It seems like it never ends. Copyrights could be really dangerous if you mess it up. So in working with Mark Fisher in particular, in drafting and bringing to life, it's actually live as a web service, the BioBrick Public Agreement, we went towards bilateral contracts and covenants not to assert any type of property right to begin to establish a public domain or public domain-like Commonwealth technology platform. I think there is ever-renewing, given the history in software, there's going to be, for the foreseeable future, an ever-renewing enthusiasm for exploring copyright. Literally every student I see coming up through iGEM, for example, who connects with property rights immediately presumes that you should be treating this stuff like code. And they're familiar with copyright being used in that context. But it's not at all obvious, and I can state this more strongly. My colleagues who are actually experts in the law actively disrecommend the application of copyright most of the time when we have these conversations. So I don't know. Actually, I'm a big fan of ship hulls and maskworks, right, and sui generis, but nobody likes that either.
All right, thank you, Drew.
Thanks, Mark.